You're welcome to this first seminar of 2025, organized by the National Institutes of Health Office of Dietary Supplements.
Text, Office of Dietary Supplements Seminar Series. ODS.od.nih.gov. Logo, National Institutes of Health, Office of Dietary Supplements.
I'm Barbara Sorkin. I co-direct the Consortium Advancing Research on Botanicals and Other Natural Products from the ODS. Before I introduce today's speaker, a few brief announcements. First, our coming attractions. Our next ODS webinar will be given on February 19th, 2025, also at 11 a.m. U.S. Eastern Time, by Dr. Elaine Langevin, Director of the NIH National Center for Complementary and Integrative Health, NCCIH. Dr. Langevin's title is Whole Person Health and the Nutrition Continuum. I hope all of you will join us again for that. You can contact the Office of Dietary Supplements at this email if you are not getting emails about ODS seminars and want to ask for the information to log in for that webinar.
Text, ODS Event Guidelines, https://ods.od.nih.gov/about/ods_event_guidelines.aspx. Attendees are expected to uphold the professional and educational purposes of meetings by respecting the rights, views, privacy, safety, and dignity of all people in their speech, postings, and other actions. Keep all comments relevant to the event topic. Refrain from engaging in harassing speech or other inappropriate behavior. Posting insulting, obscene, discriminatory, or threatening comments. Disrupting the conversation, including with spam or repetitive posts. Use of AI-based or AI-assisted note-takers. If you see or experience a violation of these guidelines, please contact ODS at ods@nih.gov or send a private message to the meeting host. Participants who engage in any of these behaviors will be given a warning from event organizers, asked to stop the behavior and be expected to stop immediately. Participants who continue to display inappropriate behavior after being given such a warning will be removed from the event.
Next, some webinar logistics. As you're already seeing here, we have muted all of you on entry in the interest of audio quality, making sure that everyone who has tuned in for this webinar is actually able to hear it. We ask that you stay muted until the final questions and answers. You do have access to the chat though, and we encourage you to put any questions you have for the speaker in the chat as they arise. If I see any that seem urgent, I'll try to get Dr. Schork's attention to ask them to interrupt him. Please also use the chat if you're having technical problems with the webinar, audio or the visual or the closed captions, and one of us here at NIH will try to help you with that. And now I am pleased to introduce, please note, no AI-assisted note takers, etc., and please be respectful of all the other attendees.
Office of Dietary Supplements Seminar Series. Any opinions expressed by ODS seminar speakers or attendees do not necessarily reflect the views of the National Institutes of Health, the Department of Health and Human Services, or the United States government. ODS.od.nih.gov. Logo, National Institutes of Health, Office of Dietary Supplements.
And with this caveat, it is my pleasure to introduce today's speaker. Dr. Nicholas Schork is a distinguished professor and co-director of the Division of Clinical Genomics and Therapeutics at TGen, a part of the City of Hope National Medical Center. He's a current member of the National Academy of Sciences, Engineering, and Medicine Special Emphasis Panel on Diet and Disease Relationships. Nick received his BA, MA, MS, and Ph.D. from the University of Michigan and is currently a principal investigator of the National Institute on Aging-sponsored Longevity Consortium, as well as the Integrative Longevity Omics Initiative, and he's a project lead for the NIA-funded Precision Aging Network Consortium. He has published more than 600 scientific articles and book chapters, mentored more than 75 trainees, has 12 patents, and has helped establish 10 companies. So we have high expectations of a great webinar, and I'm going to stop sharing my slides and hand the microphone and the projector over to you, Dr. Schork. And if you don't hear anything further from me until the end of your talk, it would be, I promise I will let you know if we're not hearing you or seeing your slides.
Great. All right. Well, it's a pleasure to be here, and I was flattered to be asked. And hopefully, what I will say in the next 45 minutes will make sense and engage you. Can everyone see my screen and it coming in clear?
Text, Genetics, Health, and Essential Nutrients. Nicholas J. Schork, Ph.D., TGen, a part of the City of Hope National Medical Center, UCSD, Scripps Research, SJHC, 45 minutes plus Q&A.
Everything is good. Go ahead, please.
All right. So my topic today is genetics, health, and essential nutrients. I'm going to focus on a few themes throughout my talk. I've kind of got my talk separated into sections. First, give a little bit of background on health interventions and geroprotector benefits. I'll define what a geroprotector is, how supplements like C15, a topic I'm going to talk about throughout today's seminar, could be a candidate geroprotector. And do want to make the statement, I don't want to offend anyone's intelligence. Some of my slides are a little basic just to set the stage for things that I'm going to talk about later.
2.
Then I'm going to talk a little bit about evolution and the polygenic nature of sustained health. As Barbara said, a lot of my funding comes through the National Institute on Aging, where we're focusing on longevity and health maintenance and kind of the factors that contribute to that, or the factors that, if modulated, could help promote or sustain health.
3.
Then I'll get into the meat of my talk, focusing on pentadecanoic acid, or C15, the first essential fatty acid discovered in about 90 years, and argue that the data suggest it has very pleiotropic effects on health, affects multiple systems, so could, in theory, be thought of as a geroprotective candidate.
4.
And then talk about its essentiality, or to be more specific, its conditional essentiality and the data behind that claim.
5.
And then close with some comments on how we can vet the geroprotective effects of supplements like C15, dietary regimens, pharmacological agents, et cetera, et cetera.
Disclosures and primary funding. Logos, Serafina Therapeutics, Net.bio, Axio Biosciences, Jump Code Genomics, Click Therapeutics, Phenomics Health, AbbVie. On the right, 6 of Dr. Schork's publications appear.
Do have a few disclosures. I am a consultant for a few different companies, including Serafina Therapeutics, which is developing C15.
Text, founder with equity and unpaid consultant.
And also I'm a consultant to the X Prize. I won't go through what the X Prize is, but they're sponsoring a huge competition to vet geroprotector candidates. The winner of the competition will get awarded upwards of $101 million. So it's a big deal. And I'm designing the trial, hence my discussion towards the tail end of my talk about the design of trials to vet geroprotectors.
1. Health interventions and geroprotector benefits.
Okay, so health interventions and geroprotector benefits, just a few background comments.
What is a geroprotector? What are good targets and MOAs? A photo of an elderly man lifting his arms appears above a comment article title, Find Drugs That Delay Many Diseases of Old Age.
Many people are interested in identifying what would amount to the fountain of youth pill or diet, et cetera, et cetera. A lot of hype and kind of craziness out there about this, but there's some serious work in this space as well.
A review in an aging and disease publication includes the title, Geroprotectors, a Unified Concept and Screening Approaches. To the right, a box contains primary selection criteria for potential geroprotectors. Text, ability to increase lifespan. Change molecular, cellular, and physiological biomarkers to a younger state or slow the progression of age-related change in these markers. Non-toxic, improve quality of life, physical, mental, emotional, etc.
Now, what is a geroprotector? People have tried to define it. Essentially, it's an intervention that has an ability to increase lifespan by changing certain molecular processes or phenomena for the better that could lead to sustained health. Of course, it's got to be non-toxic and overall improve quality of life. You could imagine engaging in interventions which keep you alive, but could compromise your ability to, say, enjoy life. That's not what a geroprotector is about.
Target the hallmarks of aging, DNA repair, mitochondrial function, stem cell depletion, etc. PMID 2374688, epigenetic, biological, clock says metrics, PMID 296434443.
Now, there's a couple of things that people have been focusing on in the context of vetting geroprotectors by asking if whatever the intervention is could actually target what are known as the hallmarks of aging, like DNA repair, mitochondrial function, a few other things I won't go into tremendous detail about, or actually manipulate or modulate an epigenetic clock. Again, I won't go through great detail about these, just say there's a growing literature on this to support the concepts of geroprotective effects and vetting geroprotectors.
Geroprotectors versus gerotherapeutics. Geroprotectors equals prevention, gerotherapeutics equals abolish disease. Rejuvenation, reprogramming, and/or regenerative? Is correcting subclinical manifestations of disease prevention of future fulminant disease or treatment of a condition?
Now, there is a distinction between geroprotectors as I've defined them and gerotherapeutics. So you could think of a gerotherapeutic as actually reversing the course of disease in someone who's manifested disease. Geroprotectors are kind of at the opposite spectrum. Think of them as preventive agents, things that sustain health. So don't necessarily correct defects that someone has accumulated over time, but actually avoid those defects or problems from occurring in the first place.
Geroprotectors, longevity-enhancing interventions with multiple targets. Table 3, number of clinical trials available by drug and age or age-related condition. On clinicaltrials.gov, condition/disease is defined as the disease, disorder, syndrome, illness, or injury that is being studied. We found approximately 12,100 clinical trials that target age or age-related diseases with 510 clinical trials aimed toward aging as a condition or disease. Of note, we have added anti-aging interventions such as CR, exercise, fasting and ketogenic diet, although they are not the focus of the review. To date, exercise is the end-aging intervention with the most ongoing clinical trials. Source: Aging Research Reviews 59, 2020.
There's a vast and growing literature on interventions that are supposed to have geroprotective properties. This is just a table from a review I was not involved in. And you can see that there's upwards of 540 clinical trials. This is actually dated, so there's more now. This was published in 2020. Trials that are focused on aging as a phenotype as opposed to diabetes or cancer or Alzheimer's disease.
An orange box highlights the table's second column, aging. In the first column, drugs, orange arrows point to interventions studied in clinical trials.
Many different compounds are being tested in bona fide registered clinical trials to see if they impact aging or aging rate or some phenomena associated with aging and longevity. Some of the biggies that you may have heard about are metformin, NAD, rapamycin, simvastatin, and then a lot of dietary interventions, including caloric restriction. I'll talk a little bit about that, fasting in other contexts, ketogenic diets, et cetera, et cetera.
Orange boxes highlight the rows for metformin and rapamycin.
Metformin and rapamycin are kind of leading the pack, a lot of attention on those. And of course, those are also being vetted for different clinical conditions and not just aging. So a lot of research going on to vet geroprotectors as I've defined them.
Text: therapeutic targeting, specific versus broad responses, polygenic perturbations. Do health interventions work on everyone?
Now, a good question is, would geroprotectors of this type work on everyone? How could you know that? What is the ubiquity of their effect? This is crucial for putting into context the public health utility of geroprotective agents of whatever sort. So you can ask questions about why won't some people respond, either with respect to specific symptoms or their overall health.
1.
Well, if someone's malnourished and you give them the pill probably isn't going to work the magic that people had thought because the person is compromised in other ways.
2.
Overeating might be another issue with respect to the geroprotective benefits of an intervention.
3.
What if an individual engages in unhealthy behaviors? Don't think taking a supplement is necessarily going to overcome the unhealthy habits that someone has engaged in.
4. Exposed to toxic substances or stressors. 5. Etc.
What if they're exposed to toxic substances? I mean, I could go on and on. The point is that many people are exposed to things or engage in behaviors that could compromise their health that a dietary supplement or a pill won't necessarily overcome. So that could then create a situation where some people are responding to the intervention and other people aren't, not necessarily because of the mechanism of action of the supplement, but because of other things plaguing the health of the people. Now, what about genetic factors?
1.
Of course, there are pharmacokinetic genetic variants influencing kind of dosing or how the body deals with a xenobiotic or something that the body is subjected to.
2.
Pharmacodynamic variations, so maybe after something hits its target, there could be genetic variations that compromise the effect of the drug.
3.
There could be system compromising monogenic insults. So if someone has, for example, a cystic fibrosis mutation, then giving them a life-enhancing pill might not be able to overcome that kind of serious genetic pathology.
4. Polygenic effects compromising various processes and the system as a whole.
And what I'll concentrate now is a polygenic variants that kind of compromise a person's health might limit the benefits that they might get from a particular geroprotective agent.
To the right, a screenshot of a clinical therapeutics paper includes the title, Clinical Pharmacokinetics and Therapeutic Drug Monitoring of Sirolimus.
To put this into context, I had mentioned rapamycin has been the leading kind of geroprotective candidate. It's been known for 30 years now that individuals show wide variation with respect to their response to rapamycin. Again, making the point that some people might not benefit as much as others to particular candidate geroprotectors, which speaks again to the ubiquity of the effect. So a geroprotective benefit must have broad pleiotropic effects to overcome many perturbations that people might have by definition. That is not an easy task.
Text, Genetics and Variation in Geroprotector Benefits, Dietary Restriction. Screenshots from a Nature article include the title, Dietary Restriction Impacts Health and Lifespan of Genetically Diverse Mice.
Now, there was a recent paper in Nature looking at the impact of dietary restriction on health and longevity in mice, outbred mice.
A response to the article appears, titled, Genetics Matters More Than Diet for Lifespan.
I was asked to comment on this paper. Just going to give you the highlights. Dietary restriction and intermittent fasting did extend female mouse lifespan. They only studied females in this paper. Many health improvements that were observed didn't actually predict lifespan extension. So one could maybe enhance health span, but not necessarily lifespan with dietary restriction. Interestingly, the strongest predictor of lifespan in these mice was resilience to stress. So nothing to do necessarily with dietary restriction, but another factor, response to stress. And genetic factors, differences between the mice, explained about 3 times more variation in lifespan than did the dietary restriction.
Text, Genetic Factors Influencing Response to Candidate Geroprotectors. 6 bar charts track metformin, rapamycin, caffeine, dasatinib, rosiglitazone, and simvastatin with respect to number of studies regarding efficacy, toxicity, dosage, metabolism/PARC, PD, and other. Source, pharmgkb.org.
Now, as I mentioned before, genetic variants can influence one's response to a candidate geroprotective agent. Here is just a table kind of listing the number of studies that have identified genetic factors that influence leading geroprotector candidates like metformin, rapamycin, et cetera, et cetera. And about 10 years ago, I had an editorial in Nature talking about how well known it is that drugs out there don't necessarily benefit everyone, and we should figure out the reasons why, so we could either tailor medicines to people to keep them healthy, or come up with drugs whose mechanism of action does have a broader and ubiquitous kind of effect.
Text, Essential Nutrient MOAs. Reveal natural targets for geroprotectors. Hypothesis, the molecular, biochemical, and physiological pathways and processes that essential nutrients nurture or impact may provide insights into the modulation of those pathways and processes through sources complementary to those currently providing these nutrients, possibly pharmacological, and lead to further improvements in health maintenance and longevity. Below the text, publication covers include vitamin C and the common cold by Linus Pauling and recommended daily allowances.
Now, here's a hypothesis I'm just going to throw out there to kind of set the tone for things I'm going to talk about later. The molecular, biochemical, and physiologic pathways and processes that are modulated by essential nutrients, right, could provide insights into other entry points to those pathways and processes, possibly ones that are amenable to pharmacological manipulation that could lead to further improvements. And as Barbara had pointed out, I have been associated with a few different groups.
Text, 2003 to 2007. On the right, Nicholas J. Schork's name highlights on a Food and Nutrition Board roster.
Back in the day, 20 years ago, I was a member of the Food and Nutrition Board for the National Academy of Sciences, kind of talking about how much of certain nutrients people should have to maintain health. So I've been steeped in this game for a long time. I'm not a nutrition expert. I'm more of a genomicist, but I was asked to participate on this panel. So I have some experience thinking about these things.
Text, 2. Evolution and the polygenic nature of sustained health.
Okay, now I want to switch gears a little bit, talk about the evolution and polygenic nature of sustained health.
Gene presence/absence and MLS across mammalian species. A Nature Communications article includes the title, "A Genomics Approach Reveals Insights into the Importance of Gene Losses for Mammalian Adaptations."
So one thing that is of interest, but not often appreciated by the scientific community, is different species not only differ with respect to the expression levels of genes or other things that could influence their phenotypic expression, their response to an intervention, et cetera, but they also differ with respect to the qualitative content of their genome, the differences in the content of the genes, not just in the expression levels of genes. So not just quantitative, but qualitative differences. And people have looked for differences between species that might explain phenotypic features they have on the basis of gene loss events.
Text, supplementary figure 12, overview of all detected gene loss events. We used a low parsimony to assign gene losses to the branches in the phylogenetic tree.
So some species actually having a gene which other species don't, that allows them to say benefit or extend their lifespan in some way. And one of the most interesting in the context of today's talk, which was discovered not so long ago, was this gulo gene, which influences vitamin C synthesis, which humans do not have and other species do have.
Text, the enzyme L-gulanolactone oxidase, the gulo gene, is involved in the endogenous synthesis of vitamin C but is not operative in humans. A Frontiers in Psychology paper includes the title, Vitamin C in Health and Disease, Its Role in the Metabolism of Cells and Redox State in the Brain.
And of course, vitamin C is an essential nutrient, but just understand humans depend on external sources of vitamin C because they lack the ability to synthesize vitamin C, as do a few other species. So identifying species differences at this qualitative level is an area of active research to try to identify things that could be.
Text, prediction of human-specific genes and sequences. And paper from Science includes the title, Integrating Gene Annotation with Ornithology Inference at Scale. Kirilenko et al., Science 380, 368, 2023.
Now, I'm engaged in this research. I'll just tell you a little bit about things we've been pursuing, working with Michael Hiller, Rusty Gage, and Kuo-Fen Lee at the Salk.
Based on TOGA ortholog inference, we collected ortholog data from the TOGA FTP site. Extracted from 63 primate assemblies. Identified 2,000+ genes having 3+ species with reported missing genes against human.
We used a tool that's been developed by Michael called TOGA to identify differences between species in gene context, looking for things that could explain phenotypic features that, say, humans have that other species do not. When we apply this to genome data, which is growing in exponential ways, lining up genomes, comparing genomes, say, in primates and humans, looking for differences, we've been able to identify regions of the genome that harbor sequence that are, say, present in humans but not present in other species.
On a chart, a blue box highlights data pertaining to great apes intact and lost genes.
Of course, raises questions about how that happened, but just suffice it to say that we've identified things, and we're not alone. Many people in the field are exploring this.
Text, examples of TOGA-derived human-specific orthologs. A chart includes genome assemblies, summaries, and gene labels. On the chart, A red box highlights a row for the FOXO3b gene.
Just give you an example. We identified a gene which was only present in humans and not in other species called FOXO3. I will not go into tremendous detail, just say we've been searching for these things to maybe explain why, say, humans have the exceptional lifespan they have relative to, say, our closestly related species like primate species, and why humans may be developing conditions that other species don't, Alzheimer's disease and some of the pathological manifestations there. So we have identified a number of sequences that look like they're unique to humans in this context.
Text, FOXO3 and longevity and the likely functional significance of FOXO3a.
Just to play out this a little bit more, FOXO3b is related to another gene, FOXO3a, which has been implicated in human longevity. I'm just showing you some papers. This gene is a true gene. It's expressed in tissues. It's not just some sequencing artifact that we identified. So we're trying to figure out what this gene does in humans and whether or not it actually contributes to some of the unique phenotypes that humans have.
Polygenic risk scores, PRS, disease associations, and genetic correlation.
Okay, so that was across species. Now I want to shift gears yet again, talk about within species variation, particularly in humans. So everyone's familiar with the concept of a genome-wide association study where you just test genetic variants for association with the phenotype, diabetes, response to a drug, whatever it is. Now it's possible to kind of identify variants that are unequivocally associated with whatever phenotype you are and just kind of sum them up and create a score. How many variants are in an individual's genome that are consistent with that person exhibiting a particular phenotype?
A graph titled Polygenic Risk Score tracks average number from 2 to 10 in intervals of 2 on the x-axis and frequency from 0 to 800 in intervals of 200 on the y-axis. Text, lower PRS equals lower risk, for example less than 3 times average risk. Higher PRS equals higher risk, for example more than 3 times average risk.
These are called polygenic risk scores, and they're being thought of as having clinical utility by many groups. I'm not going to go through this, but a paper not so long ago showed that polygenic risk scores are as predictive of many diseases as currently available monogenic tests to predict diseases. And now there are tons of repositories that just catalog all the polygenic scores that one could calculate from genome information on individuals and assign those individuals risk. Now, these polygenic risk scores have lots of issues with them. Will they work in different ancestral groups, et cetera, et cetera? Could they complement ways of assessing risk that go beyond genetics? And the answer to those questions are being debated and kind of played out in the biomedical science community.
Polygenic longevity scores, PLS, negative associations with UKB disease. The label available longevity GWAS data appears above 4 screenshots of data included in the study.
So what we did is kind of turn that concept on its head. We got genome-wide association study data from different groups focusing on genetic variants associated with longevity and health span. So instead of disease, health, and lifespan, we built polygenic longevity scores, as we call them. This was published not so long ago, and these charts might be a little hard to read, but I'll just walk you through them. Suffice it to say, we developed 11 polygenic longevity scores and then tested them for association with parental lifespan in the UK Biobank, and you can see these blue dots are all to the left of this line, suggesting that the polygenic risk scores, elevations in polygenic risk scores, kind of protected parents from an early death. So they're associated with lifespan. When we then asked if they were predictive of different diseases, so someone with a high polygenic longevity score should be less susceptible to a disease because that they didn't have the disease to compromise their lifestyle or lifespan, we found that these polygenic longevity scores were negatively correlated with a few different conditions, and this is discussed in the paper. So for example, Alzheimer's disease, coronary artery disease, heart attack, not diabetes and specific cancers, but actually were associated with resilience to death from COVID.
PLS sex disease PRS, age-specific disease prevalence. UK Biobank analysis, disease prevalence in age intervals. Yellow, 50% of the subjects having high disease PRS. Blue, 50% of subjects having low disease PRS. Red, upper 50% of disease PRSX, lower 50% of PLS. Pink, upper 50% of disease PRSX, upper 50% of PLS. Black, lower 50% of disease PRSX, lower 50% of PLS. Green, lower 50% of disease PRSX, upper 50% of PLS. Below the text, a line graph titled Alzheimer's disease appears.
Now, we also looked at these in the context of combining them with polygenic risk scores. So we could ask if people that had kind of the double whammy, a low polygenic longevity score, but a high polygenic risk score, would be not favorable to possess, and found out that yes, these polygenic longevity scores kind of interacted with polygenic risk scores. So if you did have that unfortunate double whammy, you were more susceptible to, say, diseases like Alzheimer's than you would if you just based that claim on polygenic risk scores or just polygenic longevity scores. We found that was the case for a few other diseases.
Graphs track coronary artery disease, heart attack, and type 2 diabetes.
I know I'm going through this quickly, just suffice it to say that these polygenic longevity scores were predictive of health and longevity. We validated them with a group of supercentenarians, people who lived to age 110 or more. We had genetic data from legacy cohorts and found out that these people did possess elevated polygenic longevity scores and reduced polygenic risk scores for some diseases. This is yet to be published.
Text, therapeutic targeting, specific versus broad responses, polygenic perturbations.
Okay, so we now have identified a polygenic basis for diseases and health, polygenic risk scores, polygenic longevity scores, but there's overwhelming evidence for the polygenic basis of response to health interventions. So some people benefit more from certain health interventions than others because of the polygenic load in their genome. So here's just an example.
A blood pressure flow chart splits into nodes for cardiac output and vascular resistance. At the bottom of the chart, a dashed green box highlights antihypertensive interventions. Source, BIA Education, 15, 6, 280-285, 2015, Schork, Lender, Zhao, Jacob, Genome Research, 1995.
Many people are familiar with antihypertensives. There's many out there, ACE inhibitors, beta blockers, diuretics, you name it, all have a slightly different mechanism of action. And just to show you kind of a graphical representation of the concepts I'm discussing, let's say you provided someone a beta blocker because they had an elevated heart rate, which would be a logical thing to do.
On the chart, a red X marks the pathway between beta blockers and heart. Text, X equals mutation, genetic variant, epigenetic modification or other type of perturbation compromising or enhancing a specific process or pathway.
That doesn't mean that that person doesn't have other perturbations in their system that could compromise their benefit from having a beta blocker.
6 more red X's appear on the chart.
So merely targeting a specific mutation might not, and the effects of a mutation might not lead to beneficial effects because the person could be compromised in other ways, other polygenic factors that might compromise the system a little bit.
A Cell Genomics paper includes the title, Characterizing the Genetic Architecture of Drug Response Using Gene Context Interaction Methods.
And a very recent paper by Noah Zaitlin's group looked in the UK Biobank for evidence of the polygenic nature of drug response and found, lo and behold, that there was a lot of evidence for polygenic factors influencing response to health interventions, including metformin.
Text, 3. Pentadecanoic acid, C15:0, and its pleiotropic effects on health.
Okay, so moving right along, now I want to get into C15 and pentadecanoic acid with kind of that evolution and a polygenic basis of health story behind me.
Pleiotropic. Effects of genetic variants reveal drug targets with broad effects. Sequence variant perturbations of genes and regulatory elements are natural experiments and expose what kind pleiotropic ripple effect, if any, those perturbations have on the body as a whole. Human gene variants exhibit pleiotropy to a greater degree than regulatory variants, and this has implications for drug targeting. Naturally occurring variations in genes might not be optimal in generating a functional effect, suggesting contrived modulation may be better. Pharmacological modulation of pleiotropic genes could lead to broader positive and negative effects.
But before doing that, just want to argue that there's another concept that people should be acquainted with, if not already, having to do with mechanism of action of health interventions. So one was the ubiquity effect. How many people might benefit from a health intervention? Another one is the breadth of the effect of the intervention. So does it do only one thing? So if you give someone an antihypertensive, is it only lowering their blood pressure? Is it improving their sleep? Is it causing them to lose weight? Who knows? That speaks to the breadth of the intervention. And so many people are interested in geroprotective drugs that do more than one thing to the body to sustain health. So we're all not taking, many different medicines, one for each particular phenotype. And just in the context of this argument, understand that many genes that are targeted by particular drugs or health interventions have broad pleiotropic effects. Here's just a couple of papers that point this out.
Screenshots include an annual reviews paper titled Patterns and Evolutionary Consequences of Pleiotropy and a Nature Genetics paper titled A Global Overview of Pleiotropy and Genetic Architecture and Complex Traits.
This little table shows that many genes, for example, have pleiotropic effects. These percentages are just, think of this as the number of phenotypes and things that might be influenced by modulation of a specific gene.
A paper includes the title, Machine-Guided Design of Cell Type Targeting Cis-Regulatory Elements.
Now, what's interesting is there was another very recent paper published in Nature, actually by a former grad student of mine, Ryan Toohey and Steve Riley, where they kind of predicted the genetic code for what are called cis-acting regulatory elements and found that these could be very, very cell specific. So unlike targeting a gene, targeting kind of regulatory elements that tell genes when to turn on and off and on, maybe you could induce very specific effects, whereas modulating a gene might have broad pleiotropic effects. Okay.
Text. C15's initial health benefits were observed in studies of U.S. Navy dolphins. C15 found to be associated with the health in U.S. Navy dolphins. On the left, a PLOS1 paper includes the title, Modified Fish Diet Shifted Serum Metabolome and Alleviated Chronic Anemia in Bottlenose Dolphins, Potential Role of Odd-Chain Saturated Fatty Acids.
So C15 was discovered a little while ago in a study of the Navy dolphins by my colleagues Stephanie and Eric Van Watson and team.
Clinical trial on Navy dolphins exploring modified diets.
It was found after a metabolomic screen on the healthiest Navy dolphins. So Navy dolphins is a cohort that's been studied for 30, 40 years by the Navy. They used to be used in military operations before drones and whatnot became available, but they still maintain the quality and really studied the health of these dolphins to maximize their health for use in, say, military operations at one time. So a very well-studied cohort. Now they're making the data available to academic researchers like myself. So of the healthiest dolphins, they tended to have elevated C15 levels.
10 dolphins on standard diet, 20 on modified C15 diet. Modified diet included different amounts of fish. Outcomes included changes in clinical chemistries and anemia.
That led to a clinical trial where dolphins were given C15 or not, and then looked at the health consequences of that intervention. And lo and behold, they found that the C15 intervention led to health benefits in the dolphins.
664 metabolites were measured. 25 metabolites exhibited differences between diets. 10 contained odd-chain saturated fatty acids, OCFA. C15 was the highest prioritized OCFA. Increased intake of C15 was associated with raised hemoglobin.
All this was published. The metabolomic screen to include other metabolites beyond C15, but C15 was the one that had the strongest association with the health benefits. So this was, again, discussed. This led to discussions about, well, what does C15 do? Is it an essential nutrient? What have people thought about C15 in the past, if they've looked at it in epidemiological contexts or other sorts of contexts? And here I'll talk a little bit about that. But before I go into that, just say we really studied this cohort.
Text, broad characterization of aging rates in dolphins via clinical chemistries. A paper includes the title, A 25-year longitudinal dolphin cohort supports that long-lived individuals in same environment exhibit variation in aging rates.
Here's a paper that we published just looking to see if there were differences between the healthiest dolphins and the least healthy dolphins with respect to their aging trajectories, and found evidence that that was the case, that there were groups of dolphins that appeared to age in a more healthy way than other dolphins.
A PLOS computational biology paper includes the title, "A time series analyses of blood-based biomarkers within a 25-year longitudinal dolphin cohort." A highlight on a graph is labeled with the text, "Red blood cell measures directly impact each other."
And we looked at this in different contexts, including different measurements made on the dolphins, like standard clinical chemistries that were recorded almost on a every 6-month basis over the life of the dolphins. So we could look at changes in these different clinical chemistries and found all sorts of relationships between them. Just let you know that clinical chemistries are now coming back into the radar screen of many biomedical researchers because they've been shown to predict health in a wide variety of ways. I won't go through all the literature. Just suffice it to say that what we were discovering in the dolphins had immediate kind of application in humans, and a lot of studies looking at those clinical chemistries that appeared to be consistent with healthy aging dolphins were found to be also associated with healthy aging in humans.
Text, large-scale epidemiologic studies exploring the health benefits of C15.
Okay, so what does the literature say about C15 from very large epidemiological studies that just looked at associations between, say, the levels of C15 found in the plasma or blood of individuals and health? So here is a study independent of my group. This was published before I got interested in C15. These is a meta-analysis kind of looking at the association between C15 levels and risk for type 2 diabetes. So very large cohorts, upwards of 14,000 people involved in these studies. And you can see on average, elevated levels of C15 were associated with less of a risk of type 2 diabetes. So again, this is just a compilation of independent studies, kind of a meta-analysis. Colorectal cancer, again, a big screen on a large number of people with and without colorectal cancer. And the one metabolite that showed associations with what appeared to be a resilience to colorectal cancer was C15. And that wasn't true for other metabolites right here. And I'm just giving you snapshots. The literature is full of studies like this. Here's another meta-analysis on cardiovascular disease and C15 levels. Again, a reasonably large sample size. And for the meta-analyses, resulted in pretty strong negative associations between C15 levels. Higher levels were associated with less risk for cardiovascular disease. Another interesting association, again, by groups done independently of mine, published years before I got interested. Here's one, plasma C15 levels are associated with less of a risk for metabolic syndrome like NAFLD and NASH in children. Again, one of the only metabolites to show an association. So a lot of evidence linking in really large human association studies, levels of C15, increased levels of C15 with health benefits.
C15 and the hallmarks of aging. Are the hallmarks complete? Papers appear, titled The Hallmarks of Aging. Pentadecanoic acid, C15, an essential fatty acid, shares clinically relevant cell-based activities with leading longevity-enhancing compounds, and the cellular stability hypothesis, evidence of pheratopsis and accelerated aging-associated diseases as newly identified nutritional pentadecanoic acid, C15, deficiency syndrome.
Now, I had mentioned these hallmarks of aging. I'm not going to go through them, but a good question is, how is C15 faring in the context of these hallmarks of aging when people have measured phenotypes reflective of the hallmarks of aging? I won't go through all the detail. I just say there's a couple of papers, one I was involved in with Stephanie, that kind of outlines the evidence for showing favorable impact on the hallmarks of aging for C15.
Text, C15 has been shown to stimulate many genes with health effects, stabilize cell membranes, attenuate cellular senescence, attenuate inflammaging, attenuate mitochondrial dysfunction, attenuate poor cellular signaling, activate PPAR alpha/delta and AMPK receptors, inhibit mTOR, decrease proliferation of cancer cells.
So suffice it to say, C15 appears to stimulate many genes, so it has this broad pleiotropic effect that I described, and it's been shown to be associated in a positive way with a number of different hallmarks of aging, like stabilizing cell membranes, which I'll talk about in a minute, attenuating cellular senescence, et cetera, et cetera. So again, I won't go through the details in the interest of time, just suffice it to say that there's a vast literature linking C15 levels to improvements in these hallmarks of aging.
Saturated fats and cell membrane protection. Saturated fats, no double bonds equals stable, unsaturated fats, double bonds equals unstable, for example omega-3s. A diagram represents saturated and unsaturated fatty acids in intracellular and extracellular cell membranes.
Now, I did say I want to talk about cell membranes. This is kind of an interesting thing. This is not considered one of the traditional hallmarks of aging, but maybe it should. So just for people that aren't aware of this, saturated fats like C15 have no double bonds, so they're kind of stable in cell membranes where they tend to camp out. Unsaturated fats, on the other hand, have these double bonds and are therefore unstable in your cell membranes. And the fact of the matter is you'd like your membranes to be stable. So you'd like to have a greater balance of saturated fats and unsaturated fats. This has been discussed in the literature for some time.
Text: Hulbert provided data suggesting that mammals with the most fragile, polyunsaturated fatty acids in their cell membranes had shortest lifespans. A.J. Hulbert, The Cell Membrane Pacemaker Theory of Aging, Journal of Theoretical Biology 234, No. 2, 2005, 277-288, Journal of Comparative Physiology B 184, 2014, 149-166. Short-lived mammals had higher levels of oxidative radicals caused by oxygen-driven breakdown of oily fats.
Hulbert back in the day proposed that long-lived species are likely the ones that have a favorable ratio of saturated and unsaturated fats in their cell membranes. That kind of played out with some data that was published, not completely uncontroversial, but there's some data suggesting this.
The lipid peroxidation index gives a readout of stability.
You can actually measure the stability of cell membranes with the lipid peroxidation index, which you could take. So if you wanted to know how stable your membranes were, you could look at this peroxidation index to get a phenotype. And there's a phenomena, ferroptosis, where it's kind of a programmed cell death.
Dependent on iron and characterized by the accumulation of lipid peroxides, and is genetically and biochemically distinct from other forms of regulated cell death such as apoptosis.
And this could be, say, reduced if in fact you change the ratio of saturated to unsaturated fats, say, that populate your cell membranes, and that can be done through dietary manipulations.
Thus, you want more stable fats in the membranes to promote stability and not kill off red blood cells, and this can be easily influenced by your diet and C15 levels.
So you could measure your peroxidation index pre and post, say, C15 levels or other sorts of manipulations to see if you are in fact stabilizing your red blood cells.
C15 is associated with epigenetic clocks and different health biomarkers. A paper includes the title, The Lipidome Correlates of Epigenetic Aging Across the Adult Lifespan, a population-based study.
Okay, I mentioned epigenetic clocks. I know I'm whizzing through this pretty quickly, but I know the slides will be made available so someone could do a deeper dive if interested. So here is a study on thousands of people where they measured the contemporary epigenetic clocks. I won't go through how that's measured, just know that epigenetic clocks are being studied by many people and have been shown to kind of be predictive of lifespan and kind of give an indication of how fast someone is aging. So if you have a favorable epigenetic clock, you're probably going to live a long time. If you don't, you might want to change course of action, have an intervention to slow that clock down. All sorts of controversies with epigenetic clocks that I won't go through. Suffice it to say, when you look at correlations between C15 levels and epigenetic clocks, for example, in this paper, the strongest factor, at least measured in this study, was C15 correlated with an epigenetic clock.
On a chart, a box highlights dark blue bars. Text: strongest negative correlation with epigenetic age.
So blue just means stronger correlations, not in a way that reflects positive benefits on that.
On the right, a chart appears with the title, Association of SFAs with Health Markers in 15,919 Individuals, Zhang et al. BMC Medicine, 2017, 15-203. 15,919 individuals across 8 European countries. 9 individual plasma phospholipid saturated fatty acids, SFAs. 13 lipid, hepatic, inflammation and glycemic pathway markers. Regression analysis corrected for multiple comparisons, p less than 0.00068. Standardized difference, SD unit, per 1 SD difference in each SFAs.
Another very large study looked at those clinical chemistries again that I had talked about in the context of the dolphin studies. And of all the kind of metabolites that were studied, C15 had the strongest kind of negative associations with many of these clinical chemistries. So reduced cholesterol, et cetera, et cetera. Again, from a very large study, 16,000 people. Study was not done by me. Again, it was done before I actually got interested in this. So very large studies suggesting that strong associations between C15 and many clinically relevant phenotypes.
C15 associations with lifespan in human clinical trials. Literature reports associations with human lifespan on the left and human clinical trials on the right.
Now, what about things like lifespan? So there have been some reported studies. Here is one, the suggestion of odd change saturated fatty acids like C15 related to lower total mortality in both sexes in a large study in Southeast Asia. Here's another one showing that there were differences in long-lived individuals and short-lived individuals with respect to C15 levels. Very recent clinical trials actually providing C15 to individuals in clinical context have shown the health benefits of C15. I had mentioned NAFLD and NASH. Here's 2 clinical trials right here focused on those diseases that both suggest that supplementation with C15 led to more favorable outcomes for the patients. So these were registered clinical trials focusing on the health benefits of C15, given all the evidence for C15 and its health benefits.
Text: Dietary restriction induces lithocholic acid, LCA, leading to health benefits. Papers appear, titled Lithocholic acid phenocopies anti-aging effects of calorie restriction, lithocholic acid binds TULP3 to activate sirtuins and AMPK to slow down aging, and why eating less slows aging. This molecule is key.
One little offshoot I want to talk about, this is a very recent paper published in Nature. Again, I was asked to comment on this, looking at another metabolite, lithocholic acid. So I would recommend you go look at these papers, not completely relevant to C15, but it bears on some of the statements I made before. So it's a good coincidence. 2 papers published in Nature within the month about this.
Key points: dietary restriction, DR, in mice leads to the production of LCA among other metabolites. Rigorous AMPK activation assays identified LCA as the only real stimulator of APMK. LCA binds to TULP3 to activate sirtuins and AMPK. Providing LCA to worms and flies leads to increased longevity. Note: worms and flies do not synthesize LCA, but it still activates downstream pathways such as AMPK. Mice provided LCA exhibit health improvements, but only a trend towards longer lifespan. LCA is produced by specific gut microbial species, raising questions about its essentiality. LCA has a complicated dose-response relationship and effect in humans. The methodology exploited can be used to identify other chemical.
But just to give you the highlights, what they did is they provided dietary restriction to mice and then looked at metabolites that were downstream of that intervention that changed. And one that changed in dietary restriction was LCA, lithocholic acid, and they showed that lithocholic acid actually had beneficial effects on AMP kinase activity, so a good longevity gene for those who are not interested. So then what they did was they isolated LCA and then gave it, not dietary restriction, to worms, flies, and mice, and showed lifespan improvements and improvements in health phenotypes in the mice. So again, what they did was they wanted to find kind of the consequences of dietary restriction at the molecular physiologic level. isolate those, and then consider those as interventions beyond dietary restriction and found favorable benefits for metabolites. So not unlike the C15 story where a favorable metabolite was identified and then used as an intervention.
4. C15 is a conditionally essential nutrient. Summary of studies exploring the health benefits of C15. To be published in March 2025, The Longevity Nutrient, the unexpected fat that holds the key to healthy aging, Dr. Stephanie Venn Watson, DVM, MPH.
Okay, so is C15 conditionally essential? The answer is yes. There's a couple of papers out there talking about this. There is a book going to be published by Stephanie Venn Watson in a little while like this.
The Geroprotector Evidence Scorecard. 1. Characterization in non-human, model species studies. 2. In vitro studies of activity. 3. Characterization in human exposure studies. 4. Human epidemiology studies linking levels to disease. 5. Human studies linking levels to mortality and longevity. 6. Broad diagnostic/prognostic utility studies. 7. Response and clinical phenotype monitoring studies. 8. Pharmacokinetic/pharmacodynamic studies. 9. Clinical trials in non-humans. 10. Initial clinical trials in humans. 11. Broad and context-specific clinical trials in humans. 12. Response/utility studies in different contexts, for example, ancestry.
So let's just think of the Geroprotector scorecard. So anything that might have health benefits, you want to know, is it associated with health outcomes in humans? What do mouse studies say about it? What do cell line studies say about it? And for most candidate Geroprotectors out there, there might be one line of evidence that suggests it might be favorable, but not more broadly across the different kind of contexts one would want to study a health intervention. Suffice it to say, for C15 and some of the data I showed, it hits kind of a lot of these kind of marks. So it has a very favorable scorecard suggesting that maybe humans could benefit from intake of C15, raising questions about its essentiality.
C15 has A+ score for items 1 through 4 and 8 through 10, A+/- score for items 5 through 7, and a- score for items 11 and 12. Text: C15 is a conditionally essential fatty acid. C15 is present in fatty foods, milk, butter, etc. Scientific Reports paper includes the title, Efficacy of Dietary Odd-Chain Saturated Fatty Acid Pentadecanoic Acid Parallels Broad Associated Health Benefits in Humans. Could it be essential?
So Stephanie and Ed Dennis a little while ago had a paper asking the question, is C15 essential? Given all the evidence behind it, they kind of came to the conclusion that it was. It is found in certain dietary substances like milk and butter.
Text: used as a marker of, for example, milk consumption. Found to be active and beneficial over time. First essential fatty acid in approximately 90 years. C15 is produced at low levels by gut microbial species, for example Parabacteroides. Dystacinous. Conditional essentiality. Internally produced levels do not sustain health.
In the past, C15 was used merely as a measure for the milk consumption of humans, not thought to be causal, thought only to be a marker for milk consumption, et cetera, et cetera. But since then, it meets the criteria for essentiality, or I should say conditional essentiality. This is interesting for people that are more knowledgeable in the space, but it is produced at low levels in humans by certain gut microbial species, raising questions about what is and isn't essential. If it's produced by an organism in your gut, does that really mean that there are internal levels that are created? Certainly not synthesized in the way that other species synthesize, say, vitamin C. So this paper proposed that C15 was essential.
Pentadecanoic acid, C15, clinical deficiencies, and pharmacologic aging. A paper includes the title, The Cellular Stability Hypothesis, Evidence of Ferroptosis and Accelerated Aging Associated Diseases as Newly Identified Nutritional Pentadecanoic Acid, C15, Deficiency Syndrome. Text: cellular fragility due to impaired membranes contributes to ferroptosis, leading to poor iron transport, leading further to anemia and related conditions. Global burden of disease report suggests that 1.9 billion people, 24% of the world's population, were anemic. The WHO estimates in 2019 30% of women aged 15 to 49 had anemia, as well as 40% of children 6 to 59 months old.
Now, one criteria for a nutrient being labeled as essential is that it is associated with untoward clinical consequences, like there's a syndrome, vitamin C deficiency, vitamin D deficiency. So now there's evidence that C15 deficiency leads to clinical phenotypes associated with anemia, which is a huge problem globally.
C15 deficiency leads to cellular instability and anemia, a clinical syndrome. Desired C15 levels, greater than 5 micrograms per milliliter in plasma/red blood cell membranes. 100 milligrams of C15 increases levels by approximately 1 microgram per milliliter, endogenous levels approximately 3 micrograms per milliliter.
And as I said before, the cell membranes can be influenced by C15 levels and other kind of saturated and unsaturated fats. But suffice it to say, there's evidence suggesting that C15 could ameliorate some forms of anemia, suggesting that there are clinical conditions that are associated with C15 deficiencies.
Legacy recommendations against fatty foods may have led to an inadvertent lowering of C15 levels and consequent increase in certain disease, for example, threw the baby out with the bathwater. Recent studies exploring C15 as conditionally essential. A paper includes the title, New Insights on Pentadecanoic Acid with Special Focus on Its Controversial Essentiality, a mini review.
Okay, now, since the publication of Ed and Stephanie's paper proposing C15, many groups have challenged that and called C15's essentiality into question and done a review. Here is one that reviewed all the evidence, again, independently of my lab or Stephanie's lab, and found, lo and behold, that thus considering all these elements, pentadecanoic acid, C15, is currently meeting the described criteria of an essential fatty acid. So recently published review. In fact, this was still in press at the time I pulled this down.
On the right, a pre-proof paper appears from the Journal of Nutritional Biochemistry.
Since then, the same team did a series of studies to appear in another study looking at growth in rats and other model species to argue whether or not C15 was essential.
A quote from the paper reads, "As expected, EFA-deficient rats showed growth retardation, compared to control rats. Supplementation of C15 at weaning increased early growth rate compared to deficient animals. Furthermore, the supplementation of C15 in the diet of EFA-deficient animals induced the previously undescribed synthesis of odd-chain PUFAs." These results suggest dietary C15 might counteract EFA-induced growth retardation, possibly through the synthesis of odd-chain N8 PUFAs. Yet mechanisms are to be deciphered for further validation.
Again, without going through the quote in its entirety, just say they found evidence suggesting that supplementation of C15 at weaning and whatnot influenced growth. So again, a study type to prove the essentiality of C15, and they found evidence that in fact C15 was likely essential.
Are there genetic variants that specifically influence C15 benefits? A paper includes the title, Genome-Wide Association Meta-Analysis of Circulating Odd-Numbered Chain Saturated Fatty Acids, Results from the CHARGE Consortium. Text: We found no genome-wide significant evidence that common genetic variation is associated with circulating levels of C15 or C23. Approximately 6,000 individuals were studied across multiple cohorts. Speaks to not only evolution selecting for individuals that benefit from C15 but also its likely ubiquity of effect.
Okay, now I talked about genetic variants and the ubiquity of effects of health interventions. Many people have done GWAS and say the levels of certain metabolites. So just measure a bunch of metabolites in the blood and then do a GWAS and see if there are genetic variants. What's interesting is in a study that looked at C15 with about 6,000 people, a GWAS, meta-analysis, a GWAS, where they measured other metabolites and a C15, they found that there was no genome-wide significant evidence for any variant that influenced levels of C15. That suggests that maybe there's not a high polygenic basis to response to C15, probably because evolution kind of selected out people that wouldn't respond to the external sources for C15 like milk and butter and benefit that way. Again, speaking to its essentiality and ubiquity of effect.
Pentadecanoic acid, C15, clinical deficiencies and pharmacologic aging.
Okay, now one other thing that we did is in the context of the geroprotective effects, we asked the question, well, how would C15 stack up against the leading geroprotector candidates if we subjected it to a series of assays that are meant to interrogate the activities of C15 and these other drugs?
On the right, a web screenshot includes the header, Biomap Human Biological Systems for Screening and Profiling.
So we went to a third party. This was not our lab. We paid to have these assays done by an industry standard lab called Eurofins that does large screens for a price for pharma. So we provided them C15 and asked if they compared it to rapamycin and metformin, omega-3s, other kind of leading geroprotector candidates, and got its activity profile. So this was done independently, but this paper was recently published.
Industry standard, independent third-party assay platform. 148 human primary cell-based assays stimulated to recapitulate disease states in different tissues, vasculature, immune system, skin, lung, etc. Dose-response relationships. 4,500 reference compounds for comparisons.
So without going into details, they looked at 150 or so different primary cell-based assays that were supposed to detect the favorable effects of an intervention on a number of molecular physiologic phenomena of relevance to clinical conditions, including dose-response relationships. They had upwards of 4,500 to 5,000 different compounds whose activity levels they had that they can compare C15 to.
Biomap Diversity Plus System, C15 and rapamycin. A chart contains columns for system, icon, cell type, stimulation, disease relevance, and biomarker readouts.
I won't go through all the details. This is kind of an eye chart, but just to suffice it to say, these are some of the systems that the biomap procedure kind of interrogated. And here are the systems that rapamycin showed a dose response effect to. And here are the ones that C15 did. It turns out at the end of the day that C15 had slightly more activities, favorable activities in these cell-based systems than rapamycin. So we argued, well, if people think of rapamycin as a reasonable geroprotective candidate, you might want to think of C15, given how it stacks up in these objective assays. Okay.
Text, natural and synthetic C15 analogs. A scientific reports paper includes the title, Pentadecanoyl carnitine is a newly discovered endocannabinoid with pleiotropic activities relevant to supporting physical and mental health. Text, a number of metabolites related to C15 activity have been identified in isolated pentadecanoyl carnitine. Proprietary synthetic analogs of C15 have been created and their properties are being evaluated. Studies seeking to identify more precise MOAs that go beyond ranges of C15 modulation of relevant systems are underway. A focus on the systems modulated by essential nutrients is clearly a winning strategy since evolution has shaped those systems' roles in health maintenance. Can't avoid these systems no matter what MOA your geroprotector has, since compromising them leads to unhealth.
Now, I had mentioned that C15, natural product, essential nutrient you can get from external sources like milk and butter, but there are other metabolites that are associated with C15 that could induce a favorable effect that are being studied. Most of these are proprietary, but just suffice it to say people are looking at this.
5. Vetting geroprotective effects in humans.
Okay, I'm going to whiz through this last kind of part of my talk. How can we actually vet geroprotectors? So we want to show that they have broad and ubiquitous effect, so influence multiple health systems. So most clinical trials focus on a specific primary endpoint, right? Like diabetes, like blood pressure.
Human clinical biology research, interventions as perturbations to the system. A paper includes the title, Exploring Human Biology with N of 1 Clinical Trials. Text, studies of the relationships between different human phenotypes and processes can, like any experiment, exploit perturbations to the system.
But if a geroprotector is supposed to improve your blood pressure, reduce your cholesterol, make you sleep better, have those kind of geroprotective properties, then those trials must be designed to interrogate those systems. That is not the norm, because again, most interventions are associated with a particular primary endpoint merely to get it through kind of regulatory approval.
Health interventions are perturbations to human systems. variation in responses, causal relationships between PK-like biomarkers and responses, surrogate endpoints, prognostic versus predictive biomarkers, etc. are crucial for enabling precision medicines of all sorts. Huge distinction: single symptom control versus improving overall health. Two flowcharts represent a traditional focused trial on single indication and whole body precision trials. What works for an individual?
We've argued that this is not the way things should be done. Maybe we should look at how interventions are impacting individuals and then aggregate that data instead of focusing on one primary endpoint and then finding out that some people don't respond and then study those people. So we just say, well, let's cut to the chase. In an era of wearables and easily measured phenotypes on people, we might be able to do fairly sophisticated studies on the broad breadth of effects of health interventions.
Text: single case experimental designs, SCEDs, and aggregated SCEDs. On the left, a graphic titled Interrupted Time Series, AB, SCED appears. Sections broken down into before intervention, after intervention, and time appear on the x-axis and outcome measure appears on the y-axis. Text: goal: use enough measures for statistically compelling claims. Control: adjust for confounding measures to the degree possible. Meta-analysis: aggregate the results for pattern discovery.
Now, there are ways of designing these studies, N-of-1 studies, single case experimental designs, interrupted time series. I won't go through this, but if you collected a large amount of data, baseline person, then gave them the intervention and measured a large amount of data on them longitudinally, then maybe you could identify changes that you could attribute to the drug. Placebo effects, covariate effects, all those sorts of things can be accounted for in these studies. Of course, you can mix up the number of intervals you have where a person is on or not on an intervention, like a dietary supplement. These sorts of studies are not good, quite frankly, for acute conditions where you just don't have the luxury of time and testing out different things to see if they benefit the patient. But for, say, dietary manipulations for some chronic diseases, you can run these. A whole lot of literature is brewing about these, or growing about these. Just understand the benefits of engaging in these sorts of studies. You can vet interventions with deeper phenotyping. Look at the breadth of effect. You can directly test for variation in response. How different do people respond to the intervention with respect to the phenotypes you've collected? You could learn a heck of a lot of human biology about this using these sorts of study designs. And you could aggregate these kind of focused studies to look at patterns across the people that showed response versus non-response in much more sophisticated ways than you could if you only had one or another primary endpoint collected on people at, say, a single time point, pre and post intervention. And actually, by collecting all this data on people, you might learn about someone's health so you could actually benefit the patients in real time instead of asking someone to participate in a clinical trial out of an altruistic bent where maybe the results of that study could benefit future generations. For these sorts of studies where you're focusing on individual health, you could in real time obtain insights into someone's health.
Power to detect average intervention effects versus variation in effects. 640 total observations, 320 participants with 2 measurements or 10 participants with 64 measurements, etc. Interrupted time series, AB, SCED with equal numbers of observations during A and B linear model with random effects accounting for serial correlation. Two-line graphs represent qualitative differences in response. The left graph represents treatment effect but no individual variance effect, and the right graph represents variance in treatment effect, i.e. individual effect.
I won't go through all the math, just to say these studies and their power have been studied by me and other people. This just shows that if you had to choose between, say, collecting data on a large number of people, but just at a few time points or a few measures, versus collecting more data on fewer people, you'd have orders of magnitude more power to detect variation in response if you study less people, but more data points on them longitudinally. And that's true even if the response, let's say, was qualitative and not sort of quantitative.
Punchline: more observations on fewer individuals are more powerful than fewer observations on more individuals for detecting variation in treatment response. Dr. Schork advances to a slide titled Multivariate N-of-1 Designs for Whole Body Health Effects of Intervention. On the left, equations for a multivariate model with flexible covariance matrix structure appear. On the right, graphs that represent the power of different pooled tests and the number of response variable affected appear. Text: single subject analysis, short multivariate time series. 16 different pooled univariate tests across all variables, power estimated from 1,000 simulations, 5-dose design, including 0 dose order, 20413, 5 response measures, 7 measurements per dose period equals 35 temporal measurements times 5 response measures equals 175 total measurements. No covariate effects were assumed. 1,000 simulations used to estimate power.
You can also focus on other sorts of features of these studies, like if you wanted to measure more than one thing and look at the effect on multiple phenotypes, as I argued was the case or should be the case for testing geroprotectors, you could have, again, enormous power to detect those if the effect of the intervention is such that it impacts these multiple phenotypes. Won't go through it in great detail.
Punchline: multivariate tests are much more powerful than univariate tests with corrections if there is a multivariate effect.
Just say that multivariate tests in these sorts of studies can be done, and if your intervention does influence more than one phenotype, you'll have orders of magnitude power to detect that. If it doesn't and only influences one thing, then you've got a standard drug. You don't have a geroprotector.
Text: high-dimensional SCED analysis, for example EEG data, omics, surveys, etc.
You can also look at this in the context of collecting massive amounts of data from like the microbiome or omics studies.
More variables than individuals. 2-period interrupted time series, AB, SCED, 50 individuals, 10 pre/post DX equals 20 time points. 200 simulations per 100 different effect sizes. On the right, graphs A through D represent number of variables, fraction of variables not associated, correlations between variables, and serial correlations and design issues.
Again, won't go through this. You can design studies, these aggregated single case experimental designs using things like interrupted time series with great power. And you can do this to look at mediation events.
Text: Generalized Therapeutic Drug Monitoring, TDM, Studies. A paper includes the title, Emerging Therapeutic Drug Monitoring Considerations and Opportunities in Precision Medicine. Insets contain a diagram, table, calculation, and graph from the paper.
So if you believe that C15 was causing the health benefits, you could measure the levels of C15 in these studies and see if in fact it is leading to the changes. That's called mediation. These sorts of designs can be used to pull out the mediation or causal effects of the intervention products, let's say, that you're interested in.
Text: AI/ML-based learning system, feedback and real-time evolution. A flowchart outlines a patient-focused exposure intervention trials, patient-derived constructs analysis, health intervention trials, and behavioral modification trails as patient-focused SCED trials. Text: SCED equals single-case experimental design, includes event of one crossover designs.
And you can also do these studies in the context of a much broader population scale sorts of study. Don't have time to go through this, just suffice to say we're thinking about this.
Intervention matching, better monitoring devices, patient engagement strategies, biomarkers/surrogate endpoints, causal relationships.
So aggregating data from big epi studies, pinpointing what might benefit an individual, and then testing to see if those predictions worked by using individualized studies, and then feeding the results of those N of 1 or SCEDs back into the data to make more accurate prediction models.
Conclusions and discussion items.
So in closing, sustained health and longevity are complex multifactorial processes and phenotypes. Everyone on the call is aware of this. Interventions designed to prolong health and increase longevity, geroprotectors, must have this broad effect in order to work as they are designed to.
1. Able to overcome nuanced health compromising factors individuals possess or are exposed to, or 2. Must be personalized.
C15 is conditionally essential nutrient, has broad pleiotropic effects, so kind of meets some of the criteria for having this geroprotective property. The mechanism of action of C15 and all essential nutrients could be explored to uncover other targets for intervention. Think of that LCA study that I mentioned, or even the C15 screen in the dolphins. Designing studies to vet geroprotectors and their human in vivo whole body health effects can exploit these single case experimental designs. And I'll just close by saying I have had discussions over the years with people at the FDA, given talks about these designs. They're not traditional designs, but in an era of geroprotectors and precision medicine, they may have a role to play going forward in vetting health interventions. So I'll stop there and just acknowledge my lab and funding sources.
Schork Lab. Graduate Student Committees, Ali Altonkaya, ASU, Mike Kupko, UCSD, Vanessa Lee, BU. Postdocs, Janeth Dan, Nivedita Bhadra. Lab-funded faculty, Agnes Chan, Guangfa Zhang. K awardees and mentoring, Jeremy Ellman, UCSD, Lauren Brown, SDSU. Research administration Christine Powell, Katie Koch-Davey, Lauren McCulloch, Mikasa Morf, Jennifer Doherty. Funding, NIH, NIA, NCI, NSF, TGen, AWF, ACS. Thanks for comments from Dr. Laura Getz, Keith Brown, Drs. Stephanie and Eric Van Watson, Longevity Consortium colleagues, ILO colleagues, PAN colleagues, X-Prize colleagues, NIA program officers, other colleagues, etc.
So thank you very much.
Questions?
Wow, thank you, Dr. Schork. That was a whirlwind tour of lots of really interesting data and lots of back and forth in the chat. A lot of it is about where one can get one C15. But let me start with a question. And I hadn't really thought about this in the context of caloric restriction either. Would you expect a diet sufficient in C15 for the person, depending on their gut microbiota, to have long-lasting effects? Does CR have long-lasting effects, or when you become insufficient, when you go back to eating like a typical American, typically well-off American, do you lose those effects?
Yeah, that's a great question. So the long-term effects of CR haven't really been studied in humans for all the obvious reasons. It's hard to track people from age 50 until they die, who are on and off kind of dietary restriction.
Following up the people from the clinical trials.
Yes, and that has been done, and it's been shown to have some lingering effects. But if people are off the diet, then the question is, quite frankly, are the effects seen during the intervention lingering? And that's the question that I think is hard to answer. It'd be like saying, if you took your antihypertensive medicine and lowered your blood pressure, what happens to your blood pressure when you come off the medicine? Well, all evidence is your blood pressure is going to increase. So that's probably the case for some dietary interventions too. Likely the case for C15 as well, because it's essential. You can't take it at age 10 and then never worry about it again. You're probably going to have to make sure you have enough C15 in your diet to sustain healthy levels, to keep those cell membranes strong, et cetera, et cetera. But it's a great question, and one that does bear on, say, clinical trials for reasons I said before. Very, very hard logistically to do studies that track humans from birth until death, where you've given some an intervention and some not. So we have to play in this space of inferring that short-term changes are likely to have long-term benefits.
Except if you're in Iceland, maybe, right? Yeah. And I mean, are you looking at whether you can analyze data from the All of Us study?
Absolutely. So, we have pulled down, all of us, tons of data, including genetic data. As I said before, the two questions I'm most interested in are the ubiquity of effect. So if someone is going to put something in the water to replace fluoride or keep fluoride in, we want it to have ubiquitous effect, work with everybody. But we would need to study that, see who benefits and who doesn't, maybe because of a genetic predisposition not to benefit, or maybe because they have a genetic predisposition. For those people, we need to come up with other sorts of intervention. The other question is the breadth of the effect. So again, you could imagine some drugs really only doing one thing, lowering blood pressure and doing nothing else. But if we want geroprotectors, we want drugs that impact multiple health-related phenotypes, because that by definition is a geroprotector. And both of those kind of concepts, looking at variation in response and looking at breadth of effect, those aren't front and center for a lot of clinical studies, which are focused more on reaching a particular milestone with respect to, and I'm not saying this is bad, I'm just saying this is the way it is, reaching a milestone with respect to a primary endpoint, or making the assumption that your drug is going to work on everyone, but not really following up to see if that is indeed the case.
So these are as a nutrient, you would expect it to have pleiotropic effects, whole person health
Exactly.
effects. Yeah.
Exactly. And that could be studied. And if it doesn't, you could ask the reasons why. Like if it only does one thing and one thing only. I think the evidence for C15, as I showed, is it does have broad effects because it's tapping into systems that evolution has shaped over time to be health inducing when one ingests, say, the sources for these essential nutrients.
So lots of other people have questions. I don't want to take over the whole thing. Um, one of the first questions to come in was, do the genetic factors, are there epigenetic as well as genetic factors in response to geroprotectors? And I would sort of throw in to that basket, are there other gut microbiota besides the Dicotonus that can produce the the C15? And do you really need to have that in your gut microbiota, or can you get enough from diet if you're getting enough dairy?
Great questions. And these are playing out. So a lot of those studies I mentioned, with the exception of some of those older kind of epidemiological studies, are very recent. So probably raising more questions than they answer about, say, the specific species that might be involved in the production of C15. So some have been identified, as I mentioned, but there's certainly a room to explore this more. Now, with respect to whether or not someone has enough C15, as I said before, it's easy to measure. So not only is C15 sort of a marker for, say, maybe problems that lead to anemia, so you could just measure it, say, clinically like you would another clinical chemistry, but it's also the intervention. So if you measure the levels of C15 and are found to have too low levels that might compromise your cell membranes, then you could ingest more, either as a supplement or through the dietary sources. So not unlike vitamin C and other essential nutrients, it's both a marker of kind of health and the actual intervention as well. So one could see if their C15 levels are currently adequate, and there's different criteria one can use. I didn't go through these, but there's literature out there. See if you've surpassed things. If not, and you're deficient, then of course you'd want to be taking a supplement or in fact ingesting more foods that are primary sources for C15.
And very briefly, is plasma C15 a good measure of whether you're sufficient in C15?
Yes, and these studies are playing out. So as I mentioned before, given all the interest in C15's essentiality, what we would like to see is C15 levels measured along with other clinical chemistries to see if there's a deficiency there. So you could give it. Now, that takes time and energy and the right kind of clinical studies, but those are ongoing. So all evidence suggests that, yes, simple measurements of C15 could be an indicator of whether or not you're deficient in C15.
My colleague, Dr. Laverne Brown, has been patiently waiting to ask a question. Can you unmute yourself, Laverne? Great.
Thank you, Barbara. I hope this is a quick question. So you've been talking a lot about the butter, but you also mentioned that there's these gut microbiota that could be producing the C15. And so I'm wondering, is that microbiota, can't talk today, is that also relying on the gut, on the butter and milk? Or is there some other dietary intervention that can cause an uptick of that gut microbiota and therefore
Great question, because you could imagine, maybe using some probiotic, get those microbial species in, and they then produce the C15. So you don't need to take, say, a C15 supplement. Great question. I think that is one of the areas that people are researching, but not just with respect to C15, but many other kind of favorable processes are thought to be amenable to modulation of the gut microbiome and other microbiomes. But it's a great question.
Thank you.
And there's a question in the chat about whether there have been studies looking at the optimal supplement dosing and whether there are any risks of, exceeding a safe level.
Yep. So a lot of these are discussed. There is a website, discoverc15, .com, I think it is. I can provide that in the chat if you want to put it along with my talk. I don't think I included in my slides, but discusses this and what levels might be healthiest and whatnot, and how many food sources one would need to obtain adequate levels, or what sources for supplements. Like Serafina has a supplement I had mentioned. I was a founder, though I'm not paid by them. They are producing a C15 supplement, both in clinical contexts and something you could buy off the shelves of a pharmacy. So there are criteria, and measurements can be made to see if you're deficient. And again, our goal is to sort of see if we can do the right clinical studies to argue that C15 should be treated as a bona fide clinical deficiency, something that should be measured in your routine physical exam along with your other clinical chemistries. And if you're found to be deficient, then an obvious recommendation would be to modify your diet.
Yeah, and I just put that website, I found it and put it in the chat for those of you who are asking for it. We have a note from our ODS colleague, Gretchen Van Nice, that there is a dietary supplement that provides 100 milligrams per serving. Other questions? If there's anyone who has a question that I'm missing and wants to ask it, please raise your hand and I will try to find it and call on you to ask it. Oh, here's a great question. And then I think we should let everybody go because we're past the hour. Is C15, is it lipophilic? Do you get C15 in your non-fat milk as well as in your
No, that's what's interesting. And given kind of Office of Dietary Supplements and focus on nutrition, I mean, many foods that provide C15, like milk, also have unfavorable properties and unfavorable constituents. So, lactose intolerant people probably aren't going to rely on milk as a source for C15. So as we know, foods come with many different things in them, some favorable, some not. So it is the case that when you drink milk or eat butter, for example, very controversial, and there's a story about this that I'll talk about in a minute, you could be getting kind of negative things along with the positive things. And that raises questions about how you could distinguish the two. Are there food sources that are more C15 enriched without the negative factors in the food source that you're having? So think of this: for many years, many decades, there was a campaign against fats. Right? We all know this. Reduce fat in your diet. But that was making the assumption that everything in fatty diets was unhealthy. So it could be, and ultimately, if you look at the data, and there's controversy around this, did people get healthier in the era when reduced fat intake was emphasized? And again, I don't eat a whole lot of fat in my diet for health reasons, so I kind of bought into a less fatty diet is probably good. But a question should be raised as to whether we're throwing the baby out with the bathwater with respect to some healthy fats like C15. So it just raises questions about how to go about understanding the composition of foods and which factors contribute to health and which might not. And that could be, again, very personalized. So some people could benefit from C15 to a greater degree than others. We don't know, in the context of having other things in the foods that they eat.
Yeah, certainly there are arguments that there are people who process omega-3 fatty acids differently. So one last question, and we'll let you go because we're 5 minutes past the hour now. When you take an omega-3 dietary supplement, does that have C15 in it?
No. So in that paper that I had mentioned, where we compared the activity profiles, omega-3s were compared. So omega-3s did not have the activity profile of C15. So different sort of ballgame with respect to what those nutrients do to the body, omega-3s versus C15. A lot of data, published papers contrasting the two, along with other nutrients as well.
Thank you so much. And a reminder to everyone, next month's ODS webinar by Dr. Langevin will be focusing on whole person health for those of you who are interested in nutrition and whole person health. And the month after that, Dr. Chris Schmidt will be talking about N-of-1 clinical trials and his work using those. So if you want more about the kinds of trials that might be used to study these things. So Nick, thank you so much for an absolutely fabulous webinar and for giving us so much of your time.
Thanks for having me.
It's a delight to have you. Thank you so much. Thanks everybody for joining us, and see you next month, I hope.
Logo, National Institutes of Health, Office of Dietary Supplements.